All right, guys, good morning. Today we're going to do a lesson on DNA, and I was kind of hoping we'd be back in school together by the time we did this unit because, quite honestly, I feel like it is the most difficult part of the whole biology curriculum, but we'll do our best to get through it, and I'll try to make it as simple as possible, okay? All right, so by now you realize that our genes are in our chromosomes that are inside of the nucleus of our cells, okay? Humans have 46 chromosomes. Other organisms have different amounts, okay? So if you can unwind the chromosome, I want you to think it as like an old-timey phone. You know how the cords used to get so wrapped up and tangled? Think of a chromosome like that, and it is basically tightly coiled DNA. So if we could unwind it down to its basic components, we would get the characteristic double helix shape of DNA. Okay, so what we need to know for this particular unit are the structures of the DNA, how the components pair up, and there is a strict rule about that, so that shouldn't be too bad, and then how DNA actually turns itself into a particular gene that is expressed by you. Okay? So the term for DNA is actually deoxyribonucleic acid. So you can probably see why people call it DNA. All right, now we haven't gone over the 4 kinds of organic compounds yet because we're doing things kind of backwards. But DNA is a nucleic acid. That's where the Na comes from of DNA. And all that means is that it's an organic compound that stores genetic information. Okay? And the subunits of DNA are called nucleotides, and I will show you what pieces that are made of. So what I want you to think of, if you think of a pearl necklace, right, that would be the structure. What you need to make up a pearl necklace are pearls. So think of this as a subcomponent of this, okay? All right, so the structure of DNA was actually discovered by Watson, Crick, and Wilkins in the 1950s, and it's a crazy story, which I'll show you on the next screen. Okay, since it is spirally, they actually gave it the term a double helix, and then when you unwind it, it actually looks like a plain old ladder. So the 2 sides, right, which we call the backbone, which is made out of a sugar and a phosphate group, and I'll show you that in just a second. And then the rungs of the ladder, the part that you hold on to and step on, would be made of nitrogen bases, okay? All right, so here are the people who are responsible. Watson and Crick were working on the structure of DNA as a pair. He was from England, he was from the United States. He was super young, super young, a genius pretty much, okay? And then Wilkins and Franklin were working on their own. They were also trying to figure out the structure. Well, Rosalind Franklin here did X-ray crystallography for a living. And long story short, what she did was take pictures of blood or different tissue samples to get the crystallography out of it. And this is one of the images she came up with. So people were struggling, these scientists were struggling to figure out how DNA was made. Okay, so here's the crazy part of the story. Remember, these 2 are partners and these 2 are partners, okay? So the story is that Watson and Crick got a quote-unquote sneak peek of Franklin's X-ray. And as soon as they saw the X-like structure, they knew that DNA had to be double-stranded. So they figured it out immediately after seeing this, okay? So, fast forward a little bit, this guy, and this guy got the Nobel Prize for discovering DNA. Okay, so there's a lot of rumors about exactly how did they get this image, because a video says sneak peek. Well, the rumor is that Wilkins and Franklin kind of had like a falling out, and he took her picture without her permission over to these 2 guys. So then these 3 guys got the Nobel Prize for DNA, and she did not. Now, it is not because she is a woman. That's what I initially thought also. She actually died when she was like 36 or 38 years old. She had, I think it was ovarian cancer, so she must have taken way too many X-rays. So that's why she did not get the Nobel Prize, is because she was already dead. They didn't award that to people who were dead. Okay, so, but it was her X-ray that actually gave them the clear picture of what DNA would look like. Okay, so if you imagine a chromosome and we unwind it, okay, it goes down to this characteristic strand of DNA. I know you guys have seen that before. It's very characteristic. Okay, so let's talk about what that's made of. I want you to imagine millions of these on the left and millions of these on the right. The backbone of DNA, no matter if you take it out of us, a slug, a rose bush, a mushroom, it doesn't matter. It is made out of a sugar and a phosphate group. The sugar we represent with a pentagon because it has 5 carbons in it, right? Just like a 5-sided pentagon. A phosphate group is a phosphorus with a bunch of oxygens around it, so it looks more like a circle. So imagine millions of these on each side, guys, okay? That would make the sides of the ladder. And then the inside of the ladder are nitrogen bases, and there are only 4, okay? There's one called adenine, thymine, cytosine, and guanine. And honestly, you don't have to remember those names. Don't focus on them. All I need you to memorize for me are the first letters, A, T, C, G. You can study DNA without ever knowing what their real names are. I promise, okay? And then there is something called a base pair rule. They've discovered that A always pairs with T. So if you see an A on the left side of the DNA molecule, there will always be a T on the right side, and vice versa. If this were a T, this would be an A. And then C always pairs with G. So when I was in high school, how I remembered it is AT&T was the major phone company back in the 1980s. So I remembered A went with T. But a student of mine, my 4th or 5th year of teaching, came up with apple turnovers and collard greens. Okay? So A pairs with T, C pairs with G. However you can remember that, that is probably the most important thing of a biology unit. Okay? And then you have hydrogen bonds that hold those bases together. Now, everything is the same. Those 6 basic pieces, sugar, phosphate, A, T, C, G, make up every DNA molecule of every single living thing on the planet. Even bacteria, okay? So that will never change. But it is the order of the A's and T's and C's and G's that make all of us unique. So there might be a particular order of A, T, C, G to give you brown eyes or to give you freckles or whatever, okay? All right, so here's a structure of a nucleotide. Pentagon for the sugar, circle for the phosphate group, and their nitrogen base would be in there. So there are 4 nucleotides in DNA. That is it. And this is one showing the phosphate, the sugar, and a C molecule. Okay? So if you could break all this down, this is what you would see. See? C with G, T with A, and so on. All right? So remember A with T, G with C, C with G, T with A. I'm just trying to save some time here, guys. Okay? And I'm not going to build this structure just simply for essence of time. But remember, if you can think of this, exactly the same thing over here, and then the opposite letters here. So a T, an A, a C, a C, a G, a T. Okay, we'll practice that later. So just how many genes we have has always been a debate, and it still is a debate. They started thinking we had hundreds of thousands of genes in the 1900s, and their latest research, they have it down to about 21,300 genes that actually do something. A lot of our DNA actually doesn't do anything, or they haven't discovered what it does yet, which is interesting. So another interesting thing, we think we're so different from all other living things. This is about what percentages we share with other organisms according to 23andMe. So our DNA is about 98% the same as a chimp. But guys, you got to remember, 2% of 20 to 25,000 genes is still quite a bit. And that's why there are still quite a few differences, okay? We have 92% the same as a mouse, 44% as a fruit fly, and so on. So you can see why they choose mice and chimps, of course, to do research on, right? Because they're so similar to us. Okay, so guys, the next section are basically just some notes for you. And of course, I'm going to put the PDF up on Google Classroom for you. But let's just review. DNA is basically called the code of life, and once they figured that out in the 50s, it was a really big deal. Initially, they only gave Watson and Crick credit, and it depends on what books you look in. Some people still do. DNA codes for a gene. It has the shape of a twisted ladder or spiral staircase, but the proper term is a double helix, and the subunits that make up DNA are nucleotides. Then a nucleotide is made of a sugar, a phosphate, and a nitrogen base. And I have a picture of that one here, okay? Sugar, phosphate, nitrogen base. And remember, the nitrogen bases can be A's, T's, C's, or G's. Okay? So, it codes for inherited characteristics. Let's start down here. The sugar is deoxyribose. Phosphate group, nitrogen bases, A, T, C, G. A always pairs with T, C always pairs with G. Guys, there is no exception to that. Okay, so if you can memorize that, you'll be doing great. It's called the base pair rule, and that's all you need to know on this page for now. Okay, so let me just show you, it doesn't matter what picture you look at, they're always the same. Circles are phosphates, okay? Pentagons are the sugar, which is a deoxyribose, okay? This, since it's a G, that tells me it's a nitrogen base. And if this is circling two of them, just like if you have two shoes, we call it a pair, okay? This three-piece thing, the sugar, the phosphate, the nitrogen base, is called a nucleotide, probably the second most important term. And then what holds them together is a hydrogen bond, okay? All right, one more, just show you they're the same, right? Here's the circle, so that's a phosphate. Then I always look for the pentagon, which is the sugar, okay? There's your pair, right? There's two of them. There's three pieces. That's your nucleotide. The hydrogen bond is what holds the base pairs together. This would be your nitrogen base. And they actually call the two sides of that the backbone. So then if you guys wanted to make it really complete, okay, this would obviously be a T, right? Okay, this one would be a C. This one would be a C, and this one would be a T. Okay, it is honestly that simple. All right. Okay, we're going to skip that review part for now. Okay, now the last thing I want to talk to you about, and I'm running out of time here, is DNA replication. If you guys can remember, a replica is a copy of something, right? Like they have expensive replicas of artwork and different things to protect the originals. So in order for a cell to divide and make a new cell, you have to copy your DNA first. Okay? So that is what's called replication. I want you to think of the DNA like a zipper. So before you can get to the A, T, C, and G, which is the meat of the DNA molecule, you have to unzip it. And then there's an enzyme that does that to break the bonds. Okay? So then if there was an A on one side, a free T nucleotide is going to come attached to it. So then eventually you're going to have one new strand of DNA that is half new and half old. And I will definitely put a video of that up for you. Okay? But here's a diagram of it. And after this is where I'm going to stop for today. Okay? So here's your parent strand is what they call it. And it unzipped. Okay? And then all these free nucleotides that are made in the nucleus, they're just floating around in there all the time, attach. Okay, it's almost like if you had a necklace or something that was broken and you put new pieces in there to replace it. Okay, so you're making an exact copy of the original parent strand. That's what replication is. All right, and again, I'll put some videos up here to help you a little more. Just remember, I feel like this is the hardest unit, especially the next couple parts we get into where DNA turns into a protein. So just bear with me and do the best you can. Do some extra reading or watch some extra YouTube videos if you need to, and we'll get through it. Okay? Email me if you have any questions. Thank you.